Welcome to iBOL's webinar series, BioScan, Illuminating Biodiversity. Our webinar today is presented by Dr. Michelle van der Bank. She will talk about the National Botanical Gardens Malaise program in South Africa. Michelle is a professor of botany at the University of Johannesburg in South Africa, and she is the director of the African Center for DNA Barcoding, or the ACDB for short. The ACDB's mission is to strengthen research frameworks for international, regional, and interinstitutional cooperation in Africa within the field of DNA technology for biodiversity science. Her research group uses molecular phylogenetics, comparative analysis, and intensive field work to address questions relevant to biodiversity conservation in Africa. In early February 2022, Michelle and her team deployed 22 Malaise traps in South Africa's 11 National Botanical Gardens. Their goal is to survey terrestrial arthropod diversity for 1 year in these spectacular gardens. This program could serve as a model for botanical gardens worldwide. And now, for more details, I'm going to turn it over to Michelle.
Thank you, Dirk, for the introduction, and Hannah for organizing this webinar. Good morning, good afternoon, evening, everybody. It is a real pleasure to present to you an overview of the National Botanical Garden Malaise program in South Africa. South Africa is the land of braaivleis, rappie, sunny skies, and the vuvuzela. Also known for the Kruger National Park and the Big Five and the Small Five that you can see on the slide. However, South Africa has even more to offer thanks to its incredible diversity of landscapes. With a land mass of only 2% of the world's land surface and surrounding seas of 1.1 million square kilometers, South Africa is among the smallest of the world's 17 megadiverse countries. Terrestrial biodiversity is divided into 9 biomes, which you can see here on this picture. And there are also 3 globally recognized biodiversity hotspots in South Africa. The succulent karoo that you can see here shared with Namibia. A total of 40% of the 6,356 plant species are endemic to this region. This area is also known for its high reptile and invertebrate diversity. The Cape Floristic region, which comprises 3 main vegetation types, the well-known fynbos vegetation, the endangered renosterveld, and also the strandveld. And then the 3rd hotspot is the Maputo-land, Pondoland, Albany region, shared with Mozambique and Eswatini. This is the 2nd richest floristic region in Africa after the Cape Floristic region. And then we also have the Cape Floral Kingdom in South Africa with 9,000 plant species, of which 20% are threatened with extinction. Now, current statistics have the number of South African plant species described at more than 20,400, and the estimated number of animal species at 67,000. There is, however, limited information on invertebrates available. The last comprehensive count of insect species was published in 1995 and listed 43,565 species. This was taken from Scholes and Holmes' 1985 publication. Thus, the last multi-taxa count for insects is now nearly 40 years old and outdated. A recent publication by Myberg et al. Indicated that South Africa contributed more than 56,000 insect records to BOLD, with 9,500 identified to species level. They, however, did not include the Kruger Park Malaise project. The Kruger Malaise project was a collaboration between CBG, SANBI, and the ACDB and ran for a year in the park. 25 Malaise traps were deployed at different locations in the park, and approximately 800,000 specimens was collected during that year. Of these, 370,000 were individually analyzed, and it revealed more or less 20,000 bins, which is about 43% of the known insect fauna of Southern Africa. Now, building on the success of the Kruger Malaise project, CBG, SANBI, and the ACDB is now leading an assessment of insect diversity in South Africa's National Botanical Gardens to provide baseline data on insect community structure and species diversity in the gardens to evaluate future change, and also to provide detailed information on species distributions to help South Africa meet its obligations under the CBD. Now, South Africa has 11 national botanical gardens located in 8 of the country's 9 provinces. The only province not having a botanical garden currently is the Northwest Province. These gardens conserved around 7,500 hectares of natural vegetation. 9 are classified as conservation areas, having both cultivated and areas of natural vegetation. Hantam National Botanical Garden in the Northern Cape and Kwalera, situated in the Eastern Cape, are classified as so-called wild gardens. They don't have cultivated areas. Hantam is also known or identified as a center for long-term ecological monitoring and research by SANBI. Now, Kirstenbosch, situated at the eastern foot of Table Mountain, is our oldest garden, with Thohoyandou in the Limpopo province, the youngest, established in 2021. The gardens are representatives of 7 of the South Africa's 9 biomes. The only 2 biomes not represented are the desert biome neighboring Namibia and the Indian Ocean coastal belt that you can see here. The 3 gardens in the Western Cape, the Karoo, the Harold Porter, and the Kirstenbosch Botanical Garden, as well as the Hantam Botanical Garden, falls into the winter rainfall area in South Africa. The rest are all located in the summer rainfall area. Now, Johandre has prepared a video to showcase the different vegetation types in the garden, as well as the location of some of the Malaise traps that we have deployed. I hope you enjoy the video, and I will talk to you again after the video.
insects are the most understudied animal species.
This garden is special because it has a waterfall, of which is natural. It's unlike any other garden.
The botanical garden, we should have a record of the insects that are within our botanical garden.
So we do not only focus on plants, we've broadened our focus. And all of it obviously speaks to each other.
The vegetation type is unique because here we have a semi-forest, mountainous forest.
We host some of the largest trees in the country, and this is the only location in the country where you'll find more than 5 champion trees.
At the end of the day, it's all about getting awareness so that we can take a lead to protect the biodiversity.
So this project, I think, is mainly important for us collecting data, knowing what's really what kind of different insect species we have in the garden, as well as their distribution.
starting this project will give us that foundation of the insects and where they occur and why they occur.
Yeah, I really think insects play an important role as part of the ecosystem. We shouldn't exclude them, but rather we also need to pay attention to them.
Thank you, Joandre, for the video. You can also visit the ACDB's website for more video footage about our gardens. As the seasons change from winter to spring, spectacular flower displays can be seen in the gardens of the Western Cape and also the Northern Cape. This is, for example, what you have seen on the video, Hantam during the summer season. And this is what you can expect to experience after the winter rainfall season. We hope to capture all of this for you. Now let's get back to the deployment of the traps. The project started in January 2022. Joandre and Ross traveled just under 6,000 kilometers across the country to meet up with Teresa Setusa, Deputy Director, Zoological Research, and a friendly and helpful SANBI team at the gardens. The team is responsible to remove the catch weekly, replace the collection bottles, and also to record collection data in the sample log sheet. Team members were also trained during January in standard operating procedures, which were adapted from the Kruger Malaise project to ensure specimen preservation for sequencing analysis and high data quality. Also to permit comparison of the sites at a global scale. We want to thank Teresa and her team, because without you guys, it really would be impossible to proceed with the project. 2 Malaise traps were deployed at each of the 11 National Botanical Gardens, one in the cultivated area and one in the natural vegetation. For Hantam, which you can see at the bottom of the slide, which is a natural garden as mentioned before, both traps were deployed in natural vegetation. Again, you can visit the ACDB's website to get more information about the locality of the traps, as well as additional information on the gardens. So to recap, the Malaise traps are located in 7 of South Africa's 9 biomes. 4 of the gardens are located in winter rainfall areas. The rest all occur in the summer rainfall area. Note that Pretoria, Walter Sisulu, Lowveld, and Kwalera receive early summer rains, where Tuyandou, KZN and Free State get their rain much later in the season. And we have launched. Collection started on the 1st of February and the first samples are in the bags. Ross and Johanre will make the 6,000-kilometer trip again at the end of April to collect the samples and to send it off to CBG, where it will be processed and sequenced. We hope to give you an update on the results towards the middle of the year. Now, I want to invite you to join the Botanical Gardens program. Together, we can really make a difference. Now, the ACDB team wants to thank Paul for inviting us to be part of this exciting program, Teresa Setusa and her wonderful team, Hannah, Dirk, Chris, and Pete. And then lastly, Ross has set up a poll where you can indicate your favorite garden. He will make this available on the chat box as well as on Twitter. I thank you.
Oh, thank you, Michelle, for this great talk, for the wonderful presentation. So before I start introducing the panel here, and that is with me, so I'm not alone today, I'd like to tell everyone how we do that with questions and everything else. So the chat is open, so please use it, type in your questions, and then I will read it out for you and ask whoever is ready in the big panel here to answer them, probably even discuss among us. We'll see how that goes. You also will notice that in the chat function, there's a little poll. Please vote for your favorite South African National Botanical Garden. So there's a link to a form you can click anytime, or if you want to do it later in the meeting, you can scroll back. So it's in the chat function. So I've seen that popping up. So who is with me today? So of course there's Michelle, although I can't see her right now. It seems her video is not working, but she's there. And I have Pete Hollingsworth joining us from Edinburgh, so not necessarily from South Africa. And then we have Ross and Johanre sitting somewhere outside. I'm jealous. Looks way nicer than in our area. Ryan Rattray was supposed to be joining us. I think he has connection problems. Probably he's going to join us whenever possible and just pop in. So my question before any other questions come up, Johanre and Ross, where are you?
We are currently in Malaise in the KZN province of South Africa. We're just on field work.
So, oh, here's already people voting for it. I should check. The chat is pretty distracting here. So the region where you are, that is not necessarily has anything to do with the Malaise program there, or?
It doesn't have anything to do with this National Botanical Garden Malaise program.
Mm-hmm. All righty. So that's beautiful. You guys have way better weather than we have. Everything here is gray and rainy or snowy. It's cold again. So just one general question to you, Michelle. It's a wonderful idea to just get all these botanical gardens on site with that. How came that idea about? Where did you start? When did you start thinking, hey, that would be the perfect program to get things going here? Was it really just the Kruger National Park project that saw you, okay, that is possible, or what triggered it? I know you're a botanist. It's probably a stupid question, but still, I asked.
Okay, so basically, I think the Kruger Park started everything, and then me and Paul were chatting via email a few months ago, and he wanted us to continue doing other parks in South Africa. So the option was doing botanical gardens or doing all the national parks in South Africa. So I said, let's start with the botanical gardens because it seems easier than going for the parks, because we have a huge amount of parks in the country. But then saying that, the gardens was a challenge because South Africa is a big country. And as I showed in that one slide, Johanre and Ross had to travel 6,000 kilometers to go to all these gardens. and they can't really fly with ethanol, so they had to drive. So every 3 months they will have to do this little trip. So yes, it all started in the Kruger Park. I mean, we got fantastic results there. And also after COVID, after 2 years of being stuck in the house, not being able to go out, I thought this is now a fantastic project for the team to get out again into the field and contribute to iBOL. Obviously, this is our main objective here.
Yeah, it sounds promising.
And I can add also, and I did point that out in one of the slides also, insects are a very neglected group in South Africa. Yes, I'm a botanist, but we have quite a few botanists. We have a problem with the insects. I mean, the last count of insects was about 40 years ago. So there's really a lot of work that needs to be done on that front. And I was thinking this morning, I wonder how many species are we, or bins are we going to get with this project? For the Kruger, we got nearly 20,000. My estimate is 60,000. This is what I'm thinking, or more. So, I mean, that National Botanical Garden is just a treasure. I mean, people were starting to vote now for which is their botanical garden that they like the most. And I always liked the Lowveld, but after Johannes's video of the National, I have to say this is what I'm looking for. So yes, so it's a mix of things that got us started in it, but it's all due to Paul that invited us into the program.
That was great. So I have one question from Paul, I believe. Might it be possible to barcode the plant species in the parks?
Is this question to me now?
Whoever, yeah, probably, or the other ones.
I would love to do that. I mean, this obviously it would be great if we can do that. But obviously funding, it would be quite a bit of money to do that. Especially areas like the National, the succulent Karoo, even here in the West Coast where I'm based at the moment. I mean, every year I take the third-year students out on excursion. We see hundreds of wonderful plants, but we never collect them for barcoding because we don't have the permits. So if we can apply for permits, this would be wonderful because we're in the field. We see these plants the whole time. So. Yes, well, so I would love to do that, being a botanist. But Dirk, you say I'm a botanist. My background is actually zoology.
You see, you converted. Okay. Pete, you've done these kinds of things in botanical gardens, haven't you?
No, we're just following this with great interest and enthusiasm, and my plan is to get traps set up in the Royal Botanic Garden, Edinburgh this year, and we've just got dialogue underway about other locations within the UK. So I think it's fantastic, and they're desperately keen to join the party. So I'm afraid in the middle of this, my computer died in the middle of the video, and so I was treated to the kind of wonderful shots of the Botanic Gardens early on, and I saw the first bit, and then I missed the very last bit of the talk. So Michelle, if I kind of ask any questions and that you've covered it already, just tell me it's been dealt with. I'll tell you afterwards, right? So if I'm getting repetitive. So one thing that I'm really curious about is choosing whereabouts in the gardens to locate the traps. Did you cover that, or if not, how do you go about it?
So for most of the gardens, except the 2 gardens that don't have cultivated areas, and Ross and Johanna can speak actually about this, but we chose a spot in the cultivated area and also in the natural vegetation. And Johanna and Ross went out with the curators of the gardens, and I guess they chosen the spots. Maybe Johanna, Ross, you can tell us, how did you do it?
Is we made sure we included on every botanical garden just the input from the curator or from the team that was managing. And we made sure we put it in a good representation of the natural biome that is there, as well as in a location that doesn't have high traffic in terms of visitors. In a couple of the gardens, they were concerned that the kids might play on it during the weekend. So we did try and just take into as many things into consideration as possible.
Just a sort of kind of follow-up question again, apologies if Michelle covered this earlier, but there's this sort of trade-off between location of the traps where they're not kind of interfered with by visitors on the one hand versus the public engagement opportunity on the other hand. Is there a kind of chance or ideas to use the fact that there are huge visitor numbers to these botanic gardens to have the public engagement element of this barcoding, biodiversity and everything?
So
Definitely.
What we've done, Michelle, would you like to answer?
Ross.
No,
No,
okay.
go ahead, Ross.
So what we've done is we've spoken to the SANBI team, the interpretation team, and we would like to make sure if we can put it, or we would like to put up signs next to each or next to each trap or near each trap, just so as people pass by, they'll see they can. It's not just this. What's this trap? Let's go investigate. There is a reason why they can do further research if they would like to.
Yeah, just a small infograph and maybe a QR code linking them to the project.
Yeah.
But to add to that also, we do have quite a bit of presence on Twitter, and we're trying to sort of engage with the public through that. And I see Teresa Satusa is attending the lecture from SANBI, and I'm sure we can work with Teresa and SANBI to make it more publicly available, maybe public talks and so on, because this is really what we want to do, engage the public also in the project.
Yeah, the stuff I saw on Twitter looked great, so I was it's really interesting and engaging from this far away. And yeah, I've no idea what the combined visitor numbers are for the there's a social media thing, but thinking of the combined visitor numbers across all of those gardens, that must be fairly wide-reaching.
Yeah, no, I don't know. Maybe Teresa can say, but I know it's millions.
Wow.
Our gardens are well visited. I don't know about the Handtang though, but the rest are.
And you gave us an insight into your I mean, it's difficult asking you about your favourite botanic garden, but wondering whether Johanre and Ross, sort of having visited so many gardens, are they allowed to declare a favourite, or are they keeping that to themselves?
So I do have a favourite. Johanre, what's your favourite?
Mine's probably between Quilera and Walter Sisulu.
Whereas mine, I think at the moment is Harold Porter. But it's one of those things where each garden has something absolutely unique. Whereas if you were to say which one's the best for trees, it would change. Which one's your favorite one for flowers? So it's one of those constantly changing things. And I'm sure next time we go, it will change again.
Yeah. So I have another question, just thinking of the layout. I understand you have two different traps. One is in the cultivated area, one in the more natural one. If you say a natural, or is that really one, or is it a naturalized one? So when the park was built, it's sort of regrew and everything else, or is it really just the original when the park was founded there and just left there as a protected area? Because I'm thinking of, okay, what do we look? We're likely looking at two different insect communities. One in the cultivated might even contain plants that are not necessarily native to South Africa, I'm guessing, as most botanical gardens do that. So
yeah, so the natural vegetation is really natural. I mean, it's not planted. So it's,
yeah, that's unique.
I mean, Quilera is absolutely, I mean, you saw the video with the S3 going in there. It's absolutely thicket forest there, so natural vegetation. And what is also nice is because we are looking at all the different biomes, so I'm assuming we're going to get a lot of different insect communities in it. And within, even if, say, 4 of the botanical gardens are in the savanna biome, they are in different vegetation types within the biome. So I think we're going to get really exciting stuff. I can't wait for the first results. And
great.
I mean, even the curators, I mean, when we took the project to Teresa Setusa, that talked to the curator, the head curator in Pretoria, and he went and talked to the other curators. They were so enthusiastic. The Twitter, WhatsApp messages that came through, when can we start? So South Africa is ready for this, and we're all very excited about this. We're just waiting for Pete and his team to join us now, and the rest of the world.
Well, you might have accomplished that very soon. So I have a few questions from the audience. So one would be, generally, what would it mean to have similar insect data from all botanical gardens, or as many botanical gardens around the planet?
Sorry, Dirk, what was the question?
What would it mean to have similar insect data from other botanical gardens around the world?
I mean, what we actually trying to achieve here is to get baseline data, because we don't know what we have.
Yeah.
So if we want to look at something like, for instance, effect of climate change on our insects, there's now, after that big publications that came out about insects declining, you can't really look at something like that if you don't have this baseline data available. So, I mean, in that one paper, it's actually mentioning that in South Africa, for instance, the only 2 insect groups that's very well studied is the dragonflies and the bees. The rest people don't care about, or they do care, but not in that detail. So I think if we can get this baseline data across the globe, we can really look at things like climate change, the impact of climate change.
I hear you. I guess the other one that might have studied well are mosquitoes or something like that.
Yeah.
There's a clear connection to us, so that's probably triggering a lot of the research going in there. Some other questions. One is, are you planning to develop educational materials specifically for school groups?
That was not part of the plan, but after Pete was talking again about public engagement, this is definitely something that we can do, because we have quite a big teachers group coming to ACDB on an annual basis, where we educate the teachers on barcoding and the benefits of that. So yes, we could include that together with SANBI. We could, because SANBI has an education department, so we can work with them on that.
Yeah, I mean, personally, with the programs we ran here together with schools, we had pretty good experiences with that. At some point, even engaging the schools as a field site, as an alternative to whatever you've started there. So it might be a good thing. The other question is probably one for Pete as the outsider at this point. How you would recruit participation by other gardens? How do you entice other gardens, botanical gardens around the world to actually join? What do you think? How do we get them around?
Yeah, well, one of the first ways you do it is have a wonderful pilot project with really engaging audio visuals, and I think this is a fantastic fantastic method of enticing people, and I will be asking for a copy of that video to start circulating widely after this event. And so the beauty of the world's botanic gardens is that they're very well networked. There's an organization, Botanic Gardens Conservation International, and there are all sorts of very strong bilateral relationships between botanic gardens. So there's quite an effective communication channel that's out there, and I'm really enthused about spreading this project more widely. I think that botanic gardens are fantastic magnets and traps for insect diversity, and they're fantastic magnets and traps for public engagement and people. So everything comes together in these spaces, and I think that general principle, and then the connection to public understanding of science and bringing genomics and barcoding into a garden space, all the ingredients are there. So I think, in light of this as the the groundbreaking pilot project, what we now need to do is get some set up elsewhere. There'll be a ripple effect, so we'll get this set up in the UK, and then I think we've got to start promoting this among our networks and engaging that wider group of botanic gardens. But I think it's fundamentally, you know, everything kind of seen here, it is such a it's a great initiative and a relatively you know, easy sell.
That's pretty cool. So I'm trying to keep an eye on if there are any kind of questions. There's something else I have here. Given the recent successes in harvesting eDNA from air, from whatever, right? You might have heard all that. How would you go about deploying that sort of technology and analysis across these study sites? Would you go beyond what you currently do and then just think about probably using eDNA methodology, other bulk sampling methodology?
Yeah, but we can do that because this is what was done in the Kruger Park. There was some of the samples were individually sequenced and the rest was bulged up and we did each. So yeah, we can do that. But
yeah, I guess
ideally you do want specimens, museum specimens also, preserved specimens.
Yeah, I'm guessing this is the starting point, that your reference library hinges on an individual specimen, ideally kept in a collection forever. And once that's done, go for it. Sure.
Yes. Yeah. So for us, it's still important to build that library. I mean, that paper that showed that there's, what was it, 56,000 records on bold, only 9,000 identified to species level. That was barcoded in South Africa. It shows that we have a gap here. We still need to build our reference library.
And I guess it's kind of just building on that. What I've certainly seen as important for this in the UK is as we start setting this up, is building the relationships with the natural history museums as well, so that there's you know, the traps done in the Botanic Gardens, that the barcodes are generated, but then there's a sort of good long-term repository of those specimens. So we're dialogue with National Museum of Scotland, Natural History Museum in London, thinking of those as endpoints for the specimens that we'll collect in the gardens here.
Yeah, that sounds pretty. Just naively, I've heard the comments in the video by some of the people that work at the Botanical Garden, but from their standpoint, your experience, Michelle, but also Ross and Johandro, what do you think makes them join? What is in for them? What do they consider the most interesting part of the project? We come from slightly different angles, the garden and the scientists invading with the Malaise traps, but what do you think the gardens want to have out of it?
Well, definitely, I'm not talking now about the team. I'm talking about the curators and Teresa. Obviously, they want lists of the insects that's in their gardens. This is the main goal, and for us to meet our target, South Africa. The people that's assisting us, very enthusiastic, I would say kids, they interns working in the garden. Ross and Johandre, who was dealing with them, might know why they're interested in it. One of the girls actually that they work with at the Kirstenbox Botanical Garden, she actually helped at the Kruger Park. So she was one of the people assisting there. So she was very happy to see them when they came with the Malaise traps. So maybe Johandre, you and Ross can answer that, because you were dealing with people. Why are they helping us?
So we managed to get such a wide variety of people. We managed to go from curators to interns to the average gardener being supervised by the estate manager. So each person actually had their own personal reason why they were so interested. One person might be, I want to just discover or see if there's a new species of insect. Another person wanted to see the greater trend between the cultivated and the uncultivated. Another one might want to see if there is any invasive species in the garden. So everyone had all of these elaborate reasons as to why they actually wanted to get involved, and it was depending on from what side they came from. If they were more from the garden side, then it was more garden orientated. If they were more about the biodiversity, especially at Kolera, he was very interested in the biodiversity of the garden, the biodiversity of and protecting that biodiversity. So it's such a wide range of interests that we managed to get. And the interns, this project is one of those where they actually get to present on this at, I think, the end of the year. They get to present their work that they've done on the Malaise traps. They get to present this work. So we're also trying to engage with the interns, the students, give them a couple of pictures or give them a little bit of a background about what happens. And yeah.
Overall, it's mostly people having a lack of knowledge on the insects, and everybody just wants to like sponge up more information regarding the topic.
What is wonderful to see also is every week when they go and collect the samples, they send little pictures to Andre, which we will put on Twitter. But they're very excited, they're engaging the whole time. So it's actually wonderful to see how people are engaging in the project.
That's great. Sounds like the perfect way of interacting with everyone there, not only visitors, everybody who's related to the park in one way or the other. And yeah, hopefully, by the way of the first thing that would pop in my mind when I asked the question is that they might be interested in see if there's any invasive, any pest species they don't like to have there. And this is one means of finding them probably quicker. Or pollination might be a concern in some parks.
Yeah.
So that were the first things that came to my mind. But it's interesting to hear what the motivations are from all these different people and different stakeholders, if you wish. It's good to see, but it's also probably helping to entice others because now we know what might feed their interest and why they might want to join. And I guess for them, it's low maintenance, isn't it? That's the beauty about a Malaise trap.
Yes, because it's just once a week, go and sample it and lock down the information. But yeah, it's low maintenance, but the results that you're getting out of it is just
they're huge.
It's just like
enormous. It's you have to get ready for a flood of data. Just a warning for people who heard about that before.
I remember when in the Kruger Park project, Ross and Andre went and collect the samples, and I think Michelle de Souza went with them at that point. But they came back, it was summertime. Oh, those bags were so full. They were just like solid with the insects. So this is what I'm hoping we're going to get. So yeah.
Yeah, what I foresee for some of these parks, I really would find your seasonality is way more extreme than things we can in a temperate region ever observe. When you showed the image comparing two situations in the very same park, one prior to rain season, one after, that's two different worlds to me. Only the landscape had the same shape. So that must have an impact on everything associated with that flora.
I mean, during flower time, if I go out in the field here in the Western Cape, say for instance, Kiniversveld or wherever, I put tissues literally in my nose and in my ear because it's just insects everywhere. So you can just imagine that time of the year having a trap out there. So
your bottles will be full.
I hope so.
Yeah.
So Michelle, just jumping in with another question, touch earlier on non-native species that are grown in the Botanic Gardens. I mean, obviously South Africa has just got such an incredible flora that the Botanic Gardens are wonderful places to showcase it. Do you have a feeling for the relative proportion of species in the gardens that aren't from South Africa? I know that'll vary among gardens, but is it are most of the gardens kind of almost entirely native flora?
No, for instance, KwaZulu-Natal has a lot of non-native species because, I mean, we say in Kirstenbosch is our oldest garden, but in fact, KZN, KwaZulu-Natal, is one of the oldest gardens in the world. It only recently became a National Botanical Garden. So in those gardens, you see a lot of European trees and non-native flora. But if you move to, say, Kirstenbosch, Handam, of course, Harold Porter, Karoo, those are more natural vegetation gardens.
Okay, thank you.
Great. So there's no more questions that came up there, and I thought it's probably a good time to slowly wrap it up. And to the audience out there, tell everyone who missed it, we will have a recording up on the iBOL Consortium YouTube channel, which of course will include that one particular video in it. And I'm sure you guys have somewhere where your own video will be posted on its own, because I know, for example, Pete said, I need to show that to the people, and then they're convinced, which I agree with. So this will all be available. So my last thing to do is thank you so much, everyone. Thank you, Michelle. Thank you, Ross, Johandra, Pete, Ryan. Sorry we couldn't have you on there. That's the digital age that always has its little quirks and problems. I just saw him briefly at the beginning when we were preparing, so feel sorry that he couldn't join. And thank you to the audience for joining us. In about 4 weeks, I don't have the date right now, but we'll watch out for Twitter, any kind of social media announcements. Write down, we will have another talk. And so we will continue to do the webinar on a monthly basis. We'll have the same format. We're trying to get some panelists done talking about, and feel free to join in, ask your own questions. So thank you, everyone. Have a great day wherever you are.